Lastly, we will take a look at polysaccharides. Polysaccharides are made of many monosaccharide units linked together through glycosidic linkage. And the polysaccharides are not sweet. They also have limited water solubility. The first polysaccharides we will look at is cellulose. Cellulose can be found in cotton, wood, etc. Cellulose are made by thousands of D-glucose units linked to each other through a beta-1,4 linkage. So cellulose will be a straight chain. They are linked by those 6 membered rings together in a linear fashion. When cellulose is partially hydrolyzed, it gives cellobiose. So we've learned the disaccharide cellobiose, which has 2 glucose units linked together through beta-1,4. Now you know when you have many of cellobiose linked together, you will have cellulose. So here is the structure. So I start with the structure of cellobiose, right? Then I just put parentheses around 1 unit and make an N as the subscript, which means we have N units of this structure linked together. N could be thousands. Beta-glucosidase is the enzyme that can hydrolyze beta-1,4 linkage. And emulsin, the enzyme that we talked about before that can hydrolyze beta-1,4 linkage in cellobiose, is one type of beta-glucosidase. The next polysaccharides we will look at is starch. Starch are made of D-glucose units linked together through the alpha linkage. There are 2 types of starch. One is called amylose. The other one is amylopectin. Amylose has the alpha-1,4 linkage only, so it has a straight chain. Amylopectin has a branched chain. It has not only alpha-1,4, but also alpha-1,6 linkage. So alpha-1,4 will make a straight chain, and alpha-1,6 will make a branch out of the straight chain. When amylose is partially hydrolyzed, it gives maltose only. When amylopectin is partially hydrolyzed, we can get maltose or isomaltose. So here is an example of the structure. Okay, the first unit to the second one in this structure is an alpha-1,6, and the second to the third is an alpha-1,4. And I leave the 2 ends of the bottom 2 structure open, meaning they can link to other glucose units to form the chain. Okay, but this is the basic for a starch. Lastly, glycogen. Glycogen is the animal starch. So we have glycogen stored in our liver. Glycogen is very similar to amylopectin, which means it has a branch. It has alpha 1,4 as well as alpha 1,6 linkage. But the glycogen will be much larger than amylopectin, typically 3 times larger. All right, so these are 3 types of polysaccharides. And some things are very interesting is that the animals like horses, cows, they have the enzyme that can hydrolyze beta 1,4 linkage. But humans doesn't have that enzyme. The enzyme human has is the one that can only hydrolyze alpha 1,4 linkage. So our human cannot hydrolyze cannot digest cellulose, right? So we cannot eat paper or we cannot eat leaves, those and use those as our food. But the horses, cows, they just eat grass, they eat leaves as their food because they can digest the beta 1,4 linkage. We can only use starch as our main food. Okay, so that's something that you should look forward to in your biochemistry course.